This Week in Parasitism is brought to you by the American Society for Microbiology at microbeworld.org/TWiP. This Week in Parasitism, episode number 73, recorded on June 12th, 2014. Hey everybody, I'm Vincent Racaniello, and I am with Dickson Despommier.
Good afternoon, Vincent.
How are you, Dickson?
I'm very well, thanks. And
You're looking out the window.
I am. I'm looking at a hawk. Why is that a problem? This is a radio show.
Not a problem. You, fortunately for you, have the view out the window. My back's to the
window.
window.
You know, if you were sitting here, you would have looked at that hawk also. It was a beautiful animal.
Was it a red-tailed hawk?
I believe so.
Does it have parasites?
It had a longer wingspan than I thought a red tail should have and a shorter tail. It almost looked like an eagle, but I wasn't sure.
Would it have parasites, that bird?
Oh, but of course.
What kind?
Filarial parasites. A lot of filarial parasites.
Really? So if we took out the intestines, we would find them?
Physoloptera, as I recall the genus name.
And they are of no consequence to the bird.
Well, I wouldn't say that. I mean, it depends on the worm burden.
Where do they get it, from rodents?
Well, those are the filarial parasites are transmitted by vectors.
Do you remember the other day you saw one carrying a pigeon?
Or no, we saw it carrying
a rat. Sorry, it was
we
a
saw
rat.
The tail of the rat hanging down from the talons of the red tail.
It flew right by the window.
It was amazing.
So it would get some filaria from that rat,
No.
I guess.
No.
Never?
No. Filarial parasites are carried by things like midges and mosquitoes and things like that.
What would it get from the rat?
Oh, I think a full belly.
Yeah, I was just going to say that. No parasites?
I don't think so.
The rat has no parasites in rats?
Oh, no, there are tons of parasites.
What kind?
Sure. There are blood parasites or trypanosome-like parasites.
In the U.S.?
Oh, yeah. Absolutely.
So if we sampled some subway rats, we'd find some trypanosomes?
Trypanosome lewisii is not an uncommon parasite in rats.
The world around us is surrounded by parasites.
Well, it's part of life.
And that's why we do this show.
That's true.
Which we slipped a week here because of you. It's your fault.
It's not necessarily this week in parasitism. It's here we are.
What's the name of the show, Dickson?
Here we are again, parasitism.
Today we're going to do a paper that had been requested by a listener.
That's right.
However, before we do this, we have some FU.
Uh-huh.
It means follow-up, by the way.
Excellent.
Although if I were talking to you, it would mean something else.
You know what? I think you're going to have to censor that remark out of this show because this is a clean show that children
I didn't curse. We have an email from Robin.
We do.
Who writes liquids. Last time we talked about liquids and temperature, remember?
Yeah, we did.
Liquids are capable of forming a gas-liquid interface. Maintenance of the surface requires adequate pressure in the gas. Adequate in this instance means a gas pressure greater than the vaporization pressure in the liquid. The gas pressure is a sort of cork, preventing the liquid from boiling away. The vaporization pressure increases with the temperature. When the vaporization pressure equals the gas pressure, the liquid starts to boil.
Right.
In the absence of any gas pressure, a liquid-gas interface cannot be maintained. That is why astronauts' blood would boil if they are exposed to the vacuum of space.
Without the aid of a space suit, we might add.
Well, if you have a suit, then you're not exposed to the vacuum of space, right?
This is all true.
Also, in the vacuum of space, when a solid is heated to its melting point, it will convert directly into gas, the process being known as, Dickson,
sublimation.
In Earth's atmosphere, if the solid's vaporization pressure at its melting point is greater than the atmospheric pressure, it will transit directly from solid to gas, as in the case of carbon dioxide, which we have here in the laboratory in the form of...
solid.
In the form of...
dry ice.
Thank you. I got to get it twice.
Why is it called dry ice?
It's not wet.
No, because when you put your fingers on it, you stick to it. What?
Stay near the mic.
I am near the mic.
You're not. That's better.
You're always yelling at me.
I love it. You're the only person I can yell at. I have found someone to yell at.
You just think you can yell at me.
Well, I am.
Yeah,
I know it bothers you. Yesterday on the subway, you were bothered.
No, I wasn't.
Toads versus frogs.
Right.
While toads are not a taxonomic group, use of the word toad implies the exclusion of or differentiation from frogs.
Yeah.
Toads tend to be more terrestrially adapted with a dry skin, no cutaneous respiration, no webbing between the toes, and spending most of their adult lives on terra firma.
Right.
Reptiles are a paraphyletic group.
Wait a minute. What is this in response
Our
to what
last
we were talking about?
TWiP.
Yeah, but I thought we covered some of this at least.
In our last TWiP.
We're pretty knowledgeable with regards to
A group
these.
is paraphyletic if it consists of all the descendants of the last common ancestor. It gives a definition for that.
Right.
Dinosaurs are tossed into the reptilian bin, but if they had been around to think and opine about it, they may have demurred.
Exactly.
In
Plus,
addition
they were
to the
warm-blooded.
living reptiles, there are many diverse groups that are now extinct, in some cases due to mass extinction events.
Right.
He gives a whole list of things
Right.
that were extincted.
Right.
Dinosaurs are lumped with reptiles. Although the word dinosaur means terrible lizard, the name is somewhat misleading as dinosaurs are not.
So do you know who coined the
lizards.
term dinosaur?
Who did?
Richard Owen,
Was he discovered one of the first skeletons?
the director of the British Museum of
Okay.
Natural History and also the discoverer of Trichinella. We covered that on TWiP.
Yeah.
Like 10.
Why aren't you the director of some museum? Then you'd have a pulpit.
No,
You'd have a pulpit.
No, stop it. I'm
Flaunt.
not at that.
To exhibit ostentatiously or shamelessly. Flaunts his knowledge.
Right.
Flout. To show contempt for, scorn, flout the law.
Right.
I flout Dickson. You know, Dickson, we must have talked about flaunt and flout.
We must have.
Because Robin only writes these things when we have issues.
That's true.
John writes, Dear... All right, you read this one, Dickson.
I will. It says, Dear TWiP team, on the latest episode, you insinuated perhaps that the host was chosen for his looks, and why couldn't they find someone that actually studied parasites? Well, as it happens, the host, Dan Riskin, did an AMA, Ask Me Anything, on Reddit. His expertise and passion is actually bats, but he has become a science communicator full-time. Now, I'm not sure what he meant by bats.
Bats. The animals.
His
known as
expertise
bats.
is actually bats.
Mammals. The mammals known as
No,
bats.
I got it. Okay, fine. I thought he was making some kind of a disparaging remark. When asked how he ended up as a science presenter, his response was this. I was working as a scientist in a basement when the phone rang. A production company was doing a show about evolution called Evolve for the History Channel, and they needed an evolutionary biologist who was not an old man. They had gotten my name from someone who had seen me give talks at conferences. So yes, he was picked for his looks. Showbiz. Best, John.
This is Monsters Inside Us host.
Right. No, I know very well. That's why it came up, because I was the scientific advisor for that show.
Yes, and I said, how do they pick these people on their looks? And in fact,
That's
yes, they
exactly how I was picked.
did. But you know, what's wrong with old guys or old gals?
You didn't hear what I just said.
What, sorry.
I said I was picked for the same reason,
Your looks?
for my looks. Exactly right. Only in this case, bad looks.
Who's the guy in Canada? Who's the fellow in Canada? David Suzuki. He's the science communicator. He's rather old, right?
Yeah, but he's a scientist, though.
Well, apparently this fellow was a scientist. Now
He studies bats.
I'm waiting for someone to call us. But we're old men. So they don't
I am
want
an
us.
old man. I truly admit that. I'm 74 years old. That's fine.
When do you become an old man?
I think when you think you're an old man, and not before.
So you think you're an old man?
Yeah, well, I've been slowing down.
You know when I'm an old man? When I say so.
That's right.
When
No,
other
that's-
people say so, I'm not.
That's exactly right. No,
Okay.
that's true. I totally agree with you. Do you want to read another one?
We don't
Or
have
do you want
any
to
more.
go to the
I want to
video?
go to the paper. We do the follow-up first, the FU,
Okay.
and then we do a paper.
Okay. You know where that FU comes from, by the way?
Follow-up.
No. It was from The Odd Couple.
Really?
There were two characters in The Odd Couple, right? Do you know what one of their names
Felix.
was? Felix who?
Unger.
Correct. So he left his roommate a note. He was angry with him, and he says, "And you forgot to pick out the dishes and wash the F you." And the other roommate,
Yeah.
of course, thought he was yelling
Got
at him
offended,
and doing
yeah.
something nasty.
Did they pick those initials on purpose? I
Yeah,
guess.
of course.
Felix
Of
Unger.
course they did.
You know what our initials are? R&D.
R&D, Research and Development.
Yeah.
That's right.
Racaniello and Despommier.
That's good.
Isn't that good?
I like it.
The paper we have today for
Yes.
your
Yes,
scientific pleasure
that's right.
was suggested by a listener a couple of weeks ago,
Yep.
because the name of it is Trogocytosis by Entamoeba Histolytica contributes to cell killing and tissue invasion,
Correct.
by Ralston, Solga, Mackie Lawrence, Somlata, Bhattacharya and Petrie. I just noticed that Somlata is just one name sitting there by itself. There's no first name.
That's true. No, I think it's Somlata, Bhattacharya A.
No. It's a
No, you're
name
right.
on its
You're
own.
right. No, but it's 3. The same number is over both of them.
No, it's not.
Yes, it is.
Yeah, but they're from the same place. That's weird. It's a single name person. Have you ever seen that before?
Sure.
Who?
Cher,
Prince.
Anne Margaret.
Well, that's hyphenated.
That's true.
You should go by Despommier.
Sade.
Yeah, right. Anyway.
No, I shouldn't go by the name.
This is a Nature letter.
I do go by the name of.
Dickson, give us the background on Entamoeba Histolytica.
Sure,
did we do a TWiP on this? But just give us the elevator talk.
Of course we did. Sure. Okay, so there are 2 kinds of dysentery: bacterial and amoebic.
What's dysentery?
Dysentery is bloody diarrhea.
What's diarrhea?
Diarrhea
Never mind. You don't
is
have to
watery stool. No, I can define them. I'll define them. Watery stool without blood is called diarrhea.
Mm-hmm.
Watery stool with blood is called
dysentery. Have you ever had dysentery?
No, I have actually from Vibrio parahemolyticus.
If you have dysentery, should you be afraid?
Do you know what that's from?
It's a Vibrio.
Yeah, I got it by eating raw oysters in
In
New Orleans.
Where? New Orleans?
New Orleans, my hometown.
You should have put it in
I should have.
vodka.
No, I should have. I should have had oysters Rockefeller instead, which is a cooked version of that.
If you put an oyster in vodka, will it inactivate the Vibrio?
That's a good question.
Vodka is over 70% alcohol, right?
I think it probably will, but I've never tried. So at any rate, so dysentery is bloody diarrhea. And so there are 2 kinds.
Mm-hmm.
There are kinds caused by bacterial.
Like Vibrio.
Yes,
All
and
right.
by other things like Shigella too. And Salmonella.
Mm-hmm. All bacteria.
Those are bacteria. And then there is amoebic dysentery. All right. So amoebic dysentery is caused by only one species of amoeba, Entamoeba histolytica.
Only one amoeba causes dysentery?
That's correct.
Not Naegleria.
Naegleria doesn't cause dysentery. Naegleria causes tissue invasion. It's an invader. It's an invader of tissue.
It can go into your brain, right?
It can. Just like that's exactly right.
Okay. So Entamoeba histolytica.
Entamoeba histolytica is found throughout the world.
Mm-hmm.
It is transmitted from person to person by fecal contamination. And there's a resistant stage called the cyst stage of this parasite that has 8, I'll take that back, that has 4 nuclei included into an environmentally resistant shell-like structure.
Mm-hmm.
And inside of that shell-like structure, in addition to the nuclei, you have a chromatoidal bar, which represents the accumulation of all the ribosomes from the cytoplasm of the organism. And so it's a resting stage, basically. This stage is the stage that can resist the environment so that you can acquire it by fecal contamination.
Is this an acute infection?
It can be.
It doesn't persist for many, many years.
It can do that as well. It can do lots of things.
So
if you go back to the TWiP that we did on Entamoeba histolytica, you'll get the full-blown story. But so some people become carriers.
Oh, okay.
And other people become victims.
And you can die of this from dehydration, yes. I presume.
Well, no, death is probably from the extra-intestinal form of the infection.
Extra-intestinal?
Yeah. So you've got 2 different kinds.
It goes into your blood?
That's right.
So it must invade the cell, the colon wall, or the intestinal wall, as they say in Britain.
It does. And then it gets carried to the liver, and then you can get an abscess in the liver, amoebic abscess. And it's an obligate anaerobic organism from start to finish.
In your experience here in the parasitology lab, did you ever see it?
Many times.
So it's present here in the U.S.
But I'll put an asterisk next to that many times because it now turns out that there is an identical morphological equivalent called Entamoeba dispar.
Dispar, yes.
D-I-S-P-A-R, which looks exactly, for all intents and purposes, as though it is Entamoeba histolytica under the microscope. You do a Giemsa stain or a Wright stain or a trichrome stain, and you can't tell the difference between Entamoeba dispar and Entamoeba histolytica. But if the trophozoite stage, which is the other stage of this parasite, which the cyst becomes after you ingest it.
Right.
And it goes to the large intestine, and it excysts into 4 different trophozoites from a single cyst, because each trophozoite contains a single nucleus, and each cyst contains 4 nuclei. So that's the way this thing gets a leg up real quick. You swallow it, and each cyst equals 4 organisms. So one of them is bound to succeed, you see. So
it's a good strategy, right?
Not a bad life strategy if you're an invader, okay? If the organism starts to induce these, what they call flask-shaped ulcers, if you look at the large intestinal tract, for instance, of a heavily infected person, you might see a little, like an eroded area, a hole, all right? But underneath that, in the submucosa, there's these large evaginations that the organism has created by lysing its way through the tissue. And so if you cut across them on section, they look like a flask. They have a little narrow neck and they have a wide bottom. And when you look for the organism, it's not in the cell debris in the center of the flask. It's at the living edge of the flask where the cells are still alive. That's where the organisms are doing their damage. Now, this paper addresses the issue of how does this parasite cause damage to the host.
Ah, very good, Dickson.
Okay.
I understand the name Histolytica.
Right, lytics histos. It lyses tissues. That's what its name implies.
Lytic.
That's right.
You know, if you just break words up into their
It's
roots.
pretty cool when you look at it that way, right? So there's another issue here, and that is this organism does not contain any recognizable lysosomes. So how does it lyse tissues?
Does it have a nucleus, Dickson?
Of course it does. It's a eukaryotic
Does it have
parasite.
mitochondria?
No, it doesn't. It's an anaerobe. Remember, I told you it's an anaerobe. It's
So
a
no
strict
mitochondria.
anaerobe.
Strict anaerobe.
It's a strict anaerobe. It's just like Giardia. It's a strict anaerobe.
Does it have endoplasmic
By the way,
reticulum?
I didn't
Yes.
say it trivially. Giardia and E. histolytica are treated with the same drug,
Which is?
metronidazole.
Okay.
So let's go back to the life cycle, though, because it's important to realize that when the organism, just like viruses, or just like a lot of other intracellular parasites, they have to first make contact with their host cell.
Right.
And then there's an attachment phase. You like that word. I know you do because
I
you
do
use
like
it all the time.
I used to work on
In
it.
TWiP.
I used to work on it.
Well, we're going to talk about it in TWiP now, okay?
Dickson,
so this is TWiP. Remember where you are.
You probably don't know this,
Yeah.
but I used to work on a virus attachment.
Did you know?
Yeah, you don't know because you haven't read a single one of my papers, but
I looked at the titles.
did you read the articles?
Well, I got halfway through the
You
abstracts.
just looked at the pictures, right?
I looked at the pictures, right? I looked at the references. Not one of them mentioned my name, so I just stopped reading.
Listen, you're on 3 out of my 4 podcasts.
I know.
That is a better reference than any citation I could have given you.
I think this is true.
I'm honest now.
I know.
You usually know I fool around with you a lot.
Vincent.
But I am honest.
I know that you appreciate my presence.
Absolutely.
I do know this.
I
Okay,
but I still like yelling at you.
Shut up. Entamoeba histolytica needs to attach to its host cell first before it does damage. So how do you think it does that?
Via lectins.
Yes, and what do you think the basis for that is?
What do you mean the basis?
What is the actual lectin then?
On the cell?
Yeah.
Some sialic acid probably, or some other carbohydrate.
Carbohydrate.
Galactose.
There you go. It's galactose. How did you guess?
It's common.
Because we talked about this earlier.
We didn't actually. We had
And how
not.
did this get proven?
Probably by removing.
So Chinese hamster ovary cells
Yeah, you can
are a
find
good
them
target cell for this parasite.
to attach to.
Yeah, to look at the pathology and how it works. And I think that's what they used in this paper as well. So
They use jerkats,
jerkats, that's
which are
right.
human
Okay.
lymphoid cells.
Oh, okay. So but the parasite, this parasite will engulf and destroy monolayers of CHO cells.
So they have the right attachment to
They've got galactose as
Carbohydrate,
part of
yeah.
their dangling participles. That's right. Dangling, decorated proteins on the surface. So they did an experiment to prove this. Now, what would you do to prove that galactose and not some other sugar was the actual binding lectin?
I could think of 2 ways. I could remove the galactose
You
with
could
an
do that.
enzyme,
You could do
or
that.
find a CHO mutant that doesn't put galactose on the surface.
And there's a third way, too, then. It's probably the easiest way, by the way.
Put in competitor galactose
or different sugar.
Yeah, that is an easier way. You still got some science in you, Dickson.
Stop it. You can take the man out of the lab, but you can't take the lab out of the man. So they came up with media to grow these cells in that had a different lectin, a different sugar in it. You know, they used mannose and galactose and
when there was galactose, it didn't attach.
That was the only one.
So in
we called that phenomenon Daniel in the lion's den, because you put these first-class pathogens in with these very innocent little cells,
right?
And with galactose, they're as tame as lambs. They'll just sit around and probably absorb glucose from the medium and some amino acids.
Do they multiply?
I don't think so. They need to eat cells to do that. The moment you wash the media and replace it with normal, they are off to the races.
They eat the cells.
And
they multiply.
Yeah, and to watch them eat the cells is incredible, because they almost are like vacuum cleaners. They almost look as though they're sucking them up into a pore. All right, they create these little tunnels.
Yeah, they have some movies. Of that
by the way, if you
paper.
got my textbook,
why would I have that?
I don't know. It's just a random occurrence.
Okay, there it is.
I'll be right back.
This is 5th edition of Parasitic Diseases. Don't trip, Dickson.
I said if I do, I'll see you next week.
All right, now you have your book. You're in heaven, right? Look, it was written in the front. To my friend Vince. What, were you lying?
Oh, this is my other friend, Vince.
Oh. You're looking up Entamoeba?
I am.
You know, there are movies associated with this paper which show Entamoeba devouring cells. Are you listening to me?
I am, but I have to look up the articles.
Look in the table of contents, Dickson.
No, I don't do
You're just thumbing through the whole book. It's not a way to find what you're looking for. He's gone back and forth. He's thumbing through every page. He thinks he knows where it is.
No, I didn't put the electron micrograph of the amoeba ingesting cell number 2 and digesting cell number 1 at the same time. Mm-hmm. They're greedy. They're absolutely driven by greed. They don't stop until all the cells are gone.
In the tissue culture dish.
Exactly right.
Oh, that must be a cool movie to see.
Exactly right.
So this is probably what's going on in the intestine, right? Chewing up cells and making this invaginated flask-like structure.
That's correct.
Right?
So that induces the diarrheal portion of the disease first. And then when they hit blood vessels and you start to bleed through that...
No, I see. That's where the blood comes
Then you get diarrhea, and then you get dysentery.
Got it.
All right, and by the way, it can be mistaken for lots of other clinical syndromes.
Like cancer, right?
Particularly if you don't do a stool examination, you know. So if you don't do the stool exam to look for the parasites, and you assume another disease because you don't think it's amoeba, you might be tempted to say, well, you're suffering from Crohn's disease.
Or colon cancer.
Or, no, they look for colon cancer. They can look for that, and they don't see it.
Well, they would have to do a colonoscopy, which is, if they had just examined the stool, they would have seen...
Well, I'm going to tell you about a case of a woman whose entire large intestine was resected in the name of Crohn's disease. The tissue review from another hospital was the first to discover the amoeba in the tissue.
It's horrible.
She lost her entire large intestine that way.
Was that done here?
Malpractice. No, it wasn't done here. It was done at some other hospital. But
well, that's bad. They should have looked, right?
Of course they should have looked.
Should they have gotten a travel history?
Well, that's true, but you can catch an Entamoeba Histolytica right here.
All right.
In fact, one of our best slide collection amoebae was collected from an orphanage in New Jersey.
So basically, you have blood. If you present with bloody stool,
yes,
the first thing you should do is look for some parasites.
Do a culture.
Really?
Yep. Do a culture for Shigella and Salmonella, and for Vibrio parahemolyticus, and then also send it off to the parasite lab. And then, of course, what if you don't find it?
Well, then you should do a colonoscopy, right?
Well, especially if it's a chronic disease, and it's not. And what if it's not bloody diarrhea? What if it's just intermittent diarrhea, which is the most difficult one to try to pin down?
It has to be diarrhea, though.
Yeah, you have to have
not just bloody stool. It has to be
some
diarrhea.
GI problem.
So this woman but if you have Crohn's, you will have bloody diarrhea, right?
You do.
Will it be intermittent?
Yeah, it is.
But the amoebic dysentery is going to be continuous, right?
Yes, but in some cases, I mean, you have a little bit of a bout, and then your immune system kicks in, and then it comes back out again. And by the way, this parasite secretes leucosidases, which kill off the white cells that invade. The only cell it doesn't kill off right away are eosinophils. And so you can get these large lesions in the gut tract of an eosinophilic lesion called an amiboma, which looks like a bowel tumor, and for all intents and purposes behaves like a bowel tumor, except that it doesn't metastasize. And it's loaded with Charcot-Leyden crystals, which are breakdown products from the eosinophil granules, and an occasional amoeba. And you cannot diagnose that infection by looking for the organisms in stool. They're not found there. So you have to do serological tests in this case. Or you can do a PCR.
Actually, that woman will never get colon cancer.
That's true, but she'll have a colonoscopy for the rest of her life.
Well, if they left her rectum in, it's not that bad. You don't need
no, that's true.
What do you call that when you have a bag?
Colostrum.
Colostrum bag. No, not
colostomy.
Colostomy. Colostrum is
colostrum is from milk. Colostomy, excuse me.
Because people who are at risk, as defined genetically for colon cancer, sometimes they have colectomies.
That's true.
Because both my parents died of colon cancer, and sad. My father's physician once jokingly said, "You could have a colectomy." I said,
to avoid this, just get rid of it.
Some women at
Angelina Jolie got a double breast mastectomy to avoid the risk. But you know what? As it turns out, that's not such a great idea.
Well, if you have a yearly mammogram, isn't that sufficient?
It's
I shouldn't be talking about this.
No,
this is out of my league.
Controversial. It's controversial.
I don't want my colon taken out. I will have frequent
I don't blame you.
colonoscopies.
That's right.
Sure. And we'll try and detect it early.
Okay, so back to the amoeba.
However, my dad did have bloody stool, but not diarrhea, so they wouldn't have checked him for bacteria.
Right. Yeah, he had a positive guaiac test, I'm sure. They do the guaiac test on us, too, when we go in for our annual checkup.
He didn't have, it was much more than that.
Yeah.
Because he had let it go because he was a surgeon and he didn't take care of himself.
Most doctors don't. I don't know why, but that's true. They take care of everybody else but themselves.
He was dead within a year.
Wow. I'm sorry to hear that.
Again. It was a long time ago. I got over it.
No, but I don't think you fully ever get over it.
Yeah, you do.
You're still
because
talking
time puts a big distance.
No, it does, but
he's been gone for so long.
The pain of the loss is gone, but the emptiness of his presence
No, I hardly knew him.
Oh, really?
Well, he was gone a lot.
Are we talking a lot about our personal lives on the air here?
I'm sorry.
That's okay.
I'm going to cut this out.
I
will.
Yeah. Fine.
Not that I mind, but I don't think people care.
They might.
No,
they might. They'll write you letters.
I don't think that I have any pain or emptiness left. I think I'm very happy because back then I was very pained. I didn't even want to work anymore.
Of course.
And someone said to me, "Do you think your father would like it if you stopped working because he died?" I said, "Oh, yeah, that's a good point."
He would not, of course.
He would be pissed. And now that's the way I deal with loss.
Well, my father died while I was still a graduate student.
Well, that explains everything. I was actually here, 29 years old. I was just in my first year as faculty or second year.
Wow.
Anyway.
So the mechanism of pathology, the virulence factors affecting the outcome of an infection with Entamoeba Histolytica are not well understood. That's what I want to say. So we continue to work on it, and we continue to ask new questions about old issues. And one of the new questions is, do we fully understand what happens after the amoeba attaches to the cell surface of its host cell, its target cell? And the answer is, apparently we didn't know enough about it, because what I thought happened next was that the amoeba secretes a pore protein, which then punches a hole in the cell membrane of the host cell. And at the same time, there's an induction signal that goes to the amoeba to tell it to start manufacturing lysosomal enzymes. Lysosomal enzymes.
You said they don't have lysosomes.
No, I didn't. I did. I said they don't have lysosomes.
See, I'm listening to you.
They don't have lysosomes, but they have lysosomal enzymes.
Why, are they just floating around in the cytoplasm?
Well, they're not floating around. They're not there until they contact a cell.
So they're turned off. The genes are silent.
That's right. So there must be an environmental cue.
Do lysosomes get induced?
It's not
I know. I'm asking you. Do lysosomes or just the enzymes get induced?
You're asking me how lysosomes reproduce?
No, I'm not asking you how lysosomes reproduce.
Oh. What are you asking?
You say, upon contact with a cell,
Yes.
lysosomal enzymes are induced. Are
Correct.
also lysosomes induced?
No, they do not make lysosomes. They only make lysosomal enzymes.
Okay, that's what I'm asking.
Right. So the parasitophorous vacuole, which is created by the amoeba actually wrapping itself around the host cell and then ingesting it, that vacuole that's created, that's called the parasitophorous vacuole, then contains the lysosomal enzymes, which then degrades the cell and allows it to absorb the nutrients.
This is what you thought.
Well, no, that is what happens. But
Because
there's an
now
additional step.
here's
There's an
something-
additional step.
Not what's happening in this paper.
No, there's an additional step. There's an earlier step that was unnoticed.
Mm-hmm.
So this group, who specializes in the pathogenesis of Entamoeba, started to look in different ways, because we now have tools for looking at early events with regards to attachment and digestion.
What kind of tools?
Well, you have fluorescent labels, for one thing.
And good microscopy.
And superb two-photon microscopy.
And you can take movies.
And you can take movies.
Wow.
It's fantastic.
You would have liked to live in this era, wouldn't you?
Yeah, I wish I had lived in this era.
Because you had crude techniques.
I'm dead. But if I were alive, I would have loved living in this era.
Worked in this era.
I know what you meant.
Yeah.
Vincent, I know what you meant.
Well, living is working, right? Or working is
But I
living.
still am. I'm collaborating with someone in our department, as you well know.
You're just sitting there sleeping while he talks to you.
That's not true at all. No, we discuss regularly about what to do next.
I know, but you don't do any experiments yourself.
I suggest them now. I'm a sort of a consultant. That's right. I'm
It's
not a
kind of
hands-on
scary
guy.
that you found someone to do your
It's frightening. It
experiments.
is frightening, isn't it?
So they're looking at the mechanism of killing in this paper, right?
They are.
They have these wonderful experiments.
That's right.
Like you said, they can label target cells. And I use Jurkats, which are a human B cell cell line. It's actually transformed by Epstein-Barr virus.
Yep.
You can get them from people.
How about that?
And they grow in culture, and they added these Entamoeba. Where do they get the Entamoeba?
It's grown continuously in culture.
You can grow them?
CHO cells. Yeah, of course. But you need a host cell.
Really?
They have to eat something.
And you can't provide lysates of cells. That's not enough?
No, they don't grow in cell-free media, and nor do they eat bacteria, although in the paper it says their food probably is bacteria rather than the cells that they're lysing. What they're doing when they lyse cells is finding their way through the tissues on their way to save the liver.
If you grow them in culture, there are no bacteria present.
They're not.
So
It's
bogus, right?
Well, not bogus necessarily. It was just an
misstatement.
incomplete statement. I would say it's an incomplete statement.
So they label up the host cells with various dyes.
They do.
And they put in the amoeba, and you can see the dyes being
It's amazing.
nipped off.
Nipped is the word.
Is that the right word?
Yeah, nibble.
And the pieces of so in this one experiment, they labeled the cell green.
Right.
And you can see bits of green going into the amoeba.
That's right.
It's like it's being nipped off. And hence the title Trogocytosis, which is Greek for what? Nibble. Gnaw.
Needs nibble.
Nibble.
We didn't know the term before we ran across this paper. In fact, we asked one of our most esteemed colleagues if he'd ever heard of the term, and he said no. And you were surprised because this guy's heard of everything.
So I searched PubMed for Trogocytosis. There are only 100 papers.
That's amazing.
And this one being one of them.
Right, but it's common among immune cells.
Well, that's a name given by some groups to the way immune cells like B, T, and NK cells take antigen from antigen-presenting cells. So antigen-presenting cells like macrophages or dendritic cells, they patrol our bodies. And when they pick up a foreign antigen, it could be a virus, they present peptides in major histocompatibility proteins on the cell surface to these B or T cells so that they can then proliferate.
So let's get this
And some people call this trogocytosis, the taking of the antigen from the presenting cell. And I had never heard that used before in that context, Dickson, I must tell you.
Well, I also agree with you. So there are several ways of getting things across cell membranes. Let's generalize this conversation for a moment, right? So there's there's first of all passive transport. Then there's active transport. Then there's pinocytosis for larger molecules like proteins. And now we have another way of getting something across, which is not just proteins, it's aggregates of proteins in the proteome of the membranome of the cell. Trogocytosis.
So these fellows...
And then the last mechanism, of course, is phagocytosis when you eat the whole thing.
Yeah, if you took in the whole cell, it would be phagocytosis.
So that harkens back to that ad. I can't believe. You know what?
I can't believe I ate the whole thing.
I think maybe we're going to call this episode. I can't.
But
believe
they don't. They only
ate
eat bits of it.
Oh, he ended up
Dickson,
eating the whole cell.
No, but the whole story is bits. That's what Trogo is.
In the beginning bits, but immune cells stop at that point.
Yeah, they do. They don't want to eat the whole cell.
Does not.
All right, let's
stop.
see. When we get there, if you're right, then we might name it.
I just read the paper, that's all.
This group named this nibbling by the amoeba. They call it amoebic trogocytosis. So they've taken the term that's been slightly used by the immunologists.
Why do you think they had to call it amoebic trogocytosis?
I guess not to confuse people with immunological.
Because it's not the same. Because they
different.
kept eating after they nibbled.
They eat and eat and eat.
They
until
didn't
they're
know
full.
when to stop.
Somewhere we have to get a show title out of this.
I think.
They don't
I
know
can't
when to
believe
stop.
he ate the whole thing. It's pretty good.
You're so cliched.
Well, everyone understands those cliches, though. Wormholes is not a cliche?
I don't think so. What is a wormhole?
That's,
The thing out in space. The black hole? Is that a wormhole? It's different, isn't it? Robin's going to write us now.
It's a hypothetical construct.
A wormhole?
Yeah, they don't really know they exist.
All right, I don't need to go look it up.
No.
But I don't think that was so cliched. Anyway, this first figure, they show lovely micrographs.
Confocal micrographs.
They label cells with different dyes, and they show pieces getting into the amoeba. They can time it.
Right.
And they also do an electron micrograph where they have labeled the surface of the target cells with gold.
That's right.
I'm looking here to see what the gold was attached to. Let's see. Do you remember what the gold was attached to on the surface of the cell?
I don't.
Anyway, the amoeba take up the gold bits.
Right.
How about that, Dickson?
I think that's remarkable.
Live microscopy. So these cells are alive.
Right. The microscope is
Ingested
alive.
fragments contained human cell cytoplasm 90% of the time. And mitochondria 31% of the time. They take up mitochondria. Do you think they want them?
They love all of that. It's like a smorgasbord. When they eat a cell, it's like a smorgasbord. They eat all of it.
I wonder if they're using that to grow a little aerobically for a while.
No.
Why not?
They don't do that. They eat it all.
Another unexpected finding was that human cells were alive when ingestion of fragments was initiated.
It's like being eaten by a crocodile.
Cannibalism.
Like Shel Silverstein's python.
Actually, cannibalism, you don't need to be alive, right?
Hopefully not.
Wow.
You know the joke, don't you? I won't even go there. This is not a good joke. Let's
No,
keep moving.
you can be very nasty with your jokes. So these cells were initially alive, and then there was a spike in calcium, intracellular calcium levels go up within the first few minutes, and then eventually
what
the
does
cells
that tell
died.
you?
I don't know, Dickson. Tell me.
Well,
Release of calcium from
what
the ER?
regulates calcium?
Well, my limited knowledge is that most of the calcium is within the endoplasmic reticulum.
It's calcitonin.
It's released, can be released under certain conditions.
It can.
So do you think the cells are responding to the nibbling?
Oh, big
And what's
time.
the calcium going to do?
An attempt to deliver a message to the surface saying, stop.
Maybe.
I have no idea.
And then eventually they died.
they died.
Loss of plasma membrane
That's right.
integrity.
That's right.
What's the time course here, Dickson?
About 40 minutes.
Yeah, that's right. Death starts in 10 minutes or so to 50 minutes.
Right.
As early as 10 minutes, they start to die.
Yep.
It's pretty quick.
It's very quick.
I guess when you lose membrane integrity...
What's the fastest a virus can kill a cell?
20 minutes.
Is that right?
15, 20 minutes. Yeah.
Okay.
The bacteriophages can work very quickly.
Oh, I don't in a bacterial cell, but that's not what I'm referring to.
It's a virus, isn't it?
I want a human cell.
How fast?
Mm-hmm.
Well, if you put a lot of virus on cells, you can kill them within an hour, but that's not really from replication. That's from the toxic.
You overwhelm it.
Yeah, you overwhelm them. But polio can kill a cell in a couple of hours.
Okay.
But phage, 20 minutes.
No, well,
Boom.
That's
Those are viruses, Dickson.
understood.
All right. So the cells die. And then, you know what happens when the cells die?
It becomes phagocytized.
No, it says here the amoeba detach.
Well, under ordinary circumstances, they continue
But
to
it says here, listen to this. Ingestion of fragments required living cells because pre-killed human cells
Yes, that's right.
were ingested
In another way.
intact.
That's right. That's phagocytosis.
Yeah, they take
That
up
is
the whole thing.
phagocytosis.
So that's what they need to get nutrients. So first they nibble at them, kill them,
Right.
and then somehow they ingest them.
That's where they came up with the hypothesis when they got to the next set of experiments using an exteriorized gut preparation from mice. They want to know how this organism makes a flask-shaped ulcer.
Wait, before you go in there, can I just mention a few things?
Of course.
They also show that once the cells are dead, the human cells, trogocytosis stops.
Yeah.
So I guess they can sense that they've killed the cell and they stop.
Maybe that's what the calcium does. It comes in and gives the amoeba a signal.
You need 37 degrees for this to happen.
You do. That's right. It doesn't happen at 4 degrees.
And they have some drugs
That is correct.
that inhibited it.
That's exactly right.
An inhibitor of phosphatidylinositol 3 kinase,
Right.
which is a signaling molecule.
That's right.
So that tells us that signaling is involved in this. You can imagine that as soon as the amoeba contacts, so there's a lectin, which is a sugar-binding protein on the amoeba. It contacts the galactose on the cell. That must initiate signals into the amoeba saying, okay, let's get ready to
make
chew.
some lysosomal enzymes here, kids. Let's get moving.
Also, if you inhibit the microtubules, microfilaments,
with
cytochalasin, you must know what that does. That's an old-style drug.
It's very, very old.
What does it do?
It depolymerizes tubulin.
Yeah, so that inhibited trogocytosis.
That's right.
Made the cells live.
Yeah, that's right.
Isn't that interesting? So that tells you that there are rearrangements going on in the amoeba. Okay?
Right.
Then this is cool. There is a protein called amoebic C2 domain-containing protein kinase.
Right.
Which is apparently involved in this because if you produce a dominant negative form of this in the amoeba, it prevents trogocytosis.
Amazing.
So this actually tells you there's a whole signaling pathway.
There's 4 ways to prove that it's got to be trogocytosis that kills
there's
it.
a whole pathway that remains to be deciphered here.
Right, that's
a lot of research to do.
A lot of research.
I hope they can get funded for it.
Well, this is on there high on the list of NIH organisms.
Why?
Why? Because it's so ubiquitously distributed throughout the world.
They also showed, Dickson, that they ingest red blood cells.
They do. That's what they're mainly known for.
It's what they do,
In the laboratory, that's what the technician looks for under the microscope when he's got a specimen from an infected individual. They look for amoebae with red cells in the cytoplasm.
So they give them red cells, they nibble at them, and then they ingest them.
That's correct. That's their food, basically.
Why do you think they nibble first?
No idea.
Because if they inhibit nibbling...
Is it a why question? Or
I'm sorry, Dickson. What's the function of nibbling?
Thank you.
If I just rephrase it, it's still the same thing.
No, it comes out to be a different
Dickson,
question.
Because if you inhibit nibbling, they don't take up the cells.
That's correct.
So it's some kind of a signal.
They must have to kill something first.
Oh, maybe
but is a red cell alive?
Of course it is.
Really? Really?
If the chicken red cell has a nucleus, it has a nucleus.
But it's a dysfunctional nucleus. It's not a chicken nucleus. This is a human being.
I know. I'm just trying to get around your tricky questions.
I know you are. Name a mammal that has nucleated red cells. Camels.
Anyway, you asked me if a red blood cell was living.
Yes. And it's not.
No, it can't be because it can't reproduce. It doesn't have any nucleic acids.
But it has a metabolism.
Yeah, it can't evolve.
Right.
It's kind of like a virus, isn't it?
I think it's a former cell. It used to be a cell. Right?
Yeah. What's its function?
That's exactly right.
Transport oxygen.
It does a lot of things. It also transports carbon dioxide.
Why do the amoeba wish to eat red blood cells?
Because they're really good.
No. Come on, Dickson.
They're plentiful.
This is a serious podcast.
I said they're delicious and there's lots of them.
But what do they get from them? There's a lot of hemoglobin in red blood cells.
Well, guess who else eats red blood cells?
Malaria.
Thank you. But they don't. What do they
throw
eat?
away the iron part.
That's right.
Do the amoeba throw away the iron part?
No, they
too?
don't.
What is that black component made by the malaria?
It's called hematin.
And do these guys make hemozoin?
Nope.
No, they don't.
They don't. Remember, these guys are anaerobes.
All right, so let's go to your experiment. They ex vivo made mouse intestinal tissue. So you cut it out of a mouse, you wash it,
right?
Put it in
now, ordinarily, mice are not susceptible to human Entamoeba histolytica. They are not.
Meaning, if you feed them, you don't establish an infection.
If you feed the cyst to a mouse, it doesn't infect.
But if you take these intestinal tissues and you add
the trophozoites, they will invade the tissue.
But they used amoeba here in this experiment.
No. Let's back away from this.
Oh, you're backing away.
There are 2 forms of the Entamoeba histolytica organism.
Okay.
The first form is called the cyst form, has 4 nuclei. It's the infectious form of the invading amoeba.
All right.
Then there's the trophozoite form. That's the actual eating form that they've been playing with in vitro.
Okay.
So they're going to take a culture of living trophozoites and expose the surface of a mouse intestine to the trophozoites, and the trophozoites will invade those cells. So what did they find?
The amoeba traversed the crypt.
Traversed by lysing their way through or
Tissue
nibbling.
death occurred, and amoebic trogocytosis of enterocytes
I've
preceded
got the title of our
invasion.
podcast.
What's
I'm
that?
nibbling my way back to you, babe.
What's the original? What's the original? I'm working my way back to you. It's pretty funny.
We're going to nibble our way back to you, babe.
I'm nibbling my way back to you. I'm not going to put the babe in it.
Oh, I think babe.
I don't like that. It's a little insulting. I'm nibbling my way back to you.
Right. And who sang that song?
The Four Seasons.
No. Well, yeah, you're right. The Jersey Boys.
Four Seasons.
Jersey Boys.
Jersey Boys is the musical on Broadway.
No. And a new movie with Clint Eastwood. That's my pick of the week, by the way.
They made a movie of Jersey Boys? Why Clint Eastwood?
With Christopher Walken in the movie also.
Oh, this is ridiculous.
No, Clint Eastwood was the producer.
Oh, sorry. I thought he was playing Frankie
He
Valli.
made another movie called $1 Million Baby. He made another movie.
Yeah, I didn't see that. But you know what I liked? That she was in Gran Torino. Did you see it?
You're a car freak.
Did you see that movie?
No, I didn't. But he was, he was... Clint Eastwood is a bit of a rightist kind of person, I think.
Well, the movie turned out well.
It turned out well. He was against gang warfare and stuff like this.
Well, you are for it?
Shooting, of course not.
Anyway, when they add amoeba to these intestinal explants, they traverse the crypts, and the amoeba do trogocytosis, and they engulf the enterocytes, which are the cells that make up the lining of the intestine. So basically, what have they done with this experiment, Dickson?
They've moved from in vitro to in vivo, and they've tried, and I think successfully proven, that there are more steps to the pathogenesis of amoebic dysentery than meets the eye. So they've turned back our view of the way this organism behaves to the earliest stages after attachment, and they've redefined what's happening at the cellular level. They've
All right,
done
let me
a
add
very
something
fine
to
job.
that. I would say that they have shown the relevance of trogocytosis to the pathogenesis of Entamoeba histolytica
They've discovered a new mechanism by which this organism damages cells. And if we ever revise this textbook, I guarantee you that this paper will be featured in that new edition.
Yeah, they are really fascinated by this rejection of dead cell corpses. They say it makes a nutritional role for trogocytosis seem unlikely. And that's where they say bacteria may be the primary food source.
But they don't. No, wait a minute.
I don't agree with that statement.
Well, I know I don't agree with it, and I'll tell you why. Simply from a practical standpoint, we must have seen hundreds of cases of Entamoeba histolytica during my year and a half as a technician at Columbia in 1962. And there are several amoebae that you can find in stool, which you have to differentiate from Entamoeba histolytica. And one of them is Entamoeba Coli. Mm-hmm. Its cyst is much larger than E. histolytica's, and it has 8 nuclei, or more even, than Entamoeba histolytica, which only has 4. The trophozoite also has a strange-looking nucleus. Instead of having a bullseye appearance to the chromatin in the nucleus, all right, the karyosome of the Entamoeba histolytica has a central dot. Now, I can show you a picture of that in one of these photographs.
You have to tell me which figure it is so that I could put it in the show notes. Yeah.
Well, figure 12.7.
Okay.
And as you can see clearly, and it, of course, was a highly selected photograph from a lot of trophozoites that I ran across on that slide, the karyosome has a central dot. Mm-hmm. Okay. And the chromatin spokes go out evenly from it, sort of like radii. Okay. If you look at the nucleus of Entamoeba coli, which is in the back in the atlas, It's got a concentric karyosome dot. Mm-hmm. And its nucleus has clumpy chromatin, whereas the nucleus of E. histolytica has evenly distributed chromatin around the edge of the nucleus.
Right.
So on morphological grounds, you can easily distinguish them. But there's another criteria, too. If you look in the cytoplasm of Entamoeba coli, you can find Escherichia coli and other bacteria from the microbiome inside. That's what they eat. That's their food source. But if you look in the cytoplasm of Entamoeba histolytica, you don't see any. Ever. Okay? I've never, in a human specimen, on diagnosis, found E. coli or any other bacteria-like particles inside of food vacuoles, inside these amoebae. What I have seen, of course, are red cells in partial stages of digestion, which is shown also in figure 12.7. So I don't really think from an observational standpoint that if you had a culture of Entamoeba histolytica and you flooded it with a lawn of, let's say, E. coli or something like this,
wouldn't grow.
They wouldn't grow. They don't grow like that. So I don't know why they're saying that.
Is there a strain of amoeba that will infect mice?
I don't know one, but let's look up. I mean, I don't know of any. I haven't kept up with this
Because then the experiment
field.
would be to try and infect notobiotic mice.
Oh, this is very true.
And then,
in fact, Bruce Phillips, who worked at the University of Notre Dame back in the 60s, actually used germ-free guinea pigs for his model for working with Entamoeba histolytica.
So it grew in them?
It did.
Well, there you go.
It did.
Were they devoid of bacteria or just...
Oh, yeah. No, they were totally notobiotes. They were totally free of bacteria.
What I think they're saying here, actually, this is not written in a clear way.
Right.
They say a nutritional role is unlikely, but what they're saying is it probably... They think that in the structure of the intestinal epithelium, it would be hard for the amoeba to engulf a cell because it's got tight junctions and all this, but they said they nibble the cells and kind of break up the structure, and then they can engulf it. That's their idea.
My way back to you.
Maybe nibbling would be a good title. I'm nibbling my way back to you. In that case... Oh, that's what it says. That's what we decided.
That was the title.
Boy, I have a bad memory.
A terrible memory, I would say.
Thank you, Dickson.
That's okay. I'll remind you.
I'm stopping right now.
I will remind you.
Anyway, this was a good paper. Thank you.
No, thank you. You were the one that selected it.
I'm not thanking you. I'm thanking our...
Oh, I see. Thank you for working on this interesting subject and teaching us a brand new word.
I'm going to search to see if I can find the person who sent it to us, but I can't.
Oh, that's right. You selected it from an email. Exactly right.
I should really mention his name, don't you think?
Well,
I know how to do it. I can just search all of my Trogo. Can't find the person. Thank you very much for suggesting this paper because it was pretty cool.
It was very cool.
Thanks for suggesting it. Do you want to read some email, Dickson?
Sure. I'll take the first one.
No, you can't.
All right, you go.
I'm just kidding. I love bugging you. Go ahead.
Robin writes, "Greetings, Vincent and Dickson. A quick note from one of your probable many silent listeners. I am enjoying TWiV and TWiP podcasts very much. Great learning to supplement my background in ecology and public health." Oh, that's a great combination of sciences. Quick follow-up to the meaning of invasive. I think the student of ecology, Vincent, got this one right over the teacher, Dickson. I believe invasive does mean it takes over and outcompetes native species, whereas exotic is something occurring when it isn't natural. There are exotic species that are not considered invasive, although it is probably a matter of degree, since any exotic species would compete with natives for resources at some level. Perhaps that is what Dickson was implying? Well, Dickson is reading this email, and I don't remember the exactness of the arguments that we were leveling at the term, but I'll gladly ascend to the view of a bona fide ecologist, especially one with a background in public health. There's nothing more powerful than a public health person with an ecology background. That is exactly what public health is all about.
Yeah, I mean, I asked you if an invasive species takes over, and you said no, it just goes there, but this person probably knows more than you do.
Much more than I do. And in fact, but the paper that I read on the subject of whether invasive species are displacing native species or are filling a gap vacated by native species, That's probably what we were talking about.
This Robin is from Missoula, Montana.
And that's one of my favorite all-time places. You're about to go there,
I'm
aren't you,
going
Vincent?
there, Robin. I'm going to give a TWiP in Hamilton, right?
Oh, yeah.
The Rocky Mountain Laboratories
yeah.
on Friday, June 27th.
Wow. Why don't you invite her? Him.
I don't know if he or she is allowed in the facility, you know?
Well, that's kind of
It's an NIH thing.
It's a closed loop.
Next one's from Bobby.
Yep.
Dear TWiP group, first I'd like to say that I love your program. Great work. Second, I wanted to introduce you to my Parasite Case of the Week blog.
Oh, nice.
parasitewonders.blogspot.com. We just hit our 300th case.
Cool.
It's a simple blog, short cases and short answers the following week. I'd be happy to talk to you about it if you're interested. Best wishes.
Wow. We'll put a link to that, that's for sure.
We're going to have Bobby on the show.
Oh, nice.
Bobby Pritt is a director of parasitology at the Mayo Clinic.
Nice.
This is a person with knowledge, Dickson.
I see all those degrees. I'm absolutely impressed. And the Mayo Clinic is in a beautiful area, by the way. Bobby, I've actually done some trout fishing nearby where you are.
But we're going to have her on TWiP. I emailed her. She said she'd love to come on.
Oh, great.
And we can talk about what she does.
That's marvelous.
And you guys can compare notes because you used to work in a parasitology lab. And we can talk about why she started this Parasite Case of the Week, which is pretty cool.
Very, very
So everybody check it out.
Lovely. John writes, hello. iTunes said it has been almost a month since your last episode. It is spring, 15 degrees C in Massachusetts, and time for you to emerge from hibernation. Thank you, John. I wanted to call your attention to two related papers on non-human parasites. Parasites infecting non-human hosts. I was disappointed to learn from an early TWiP episode that tapeworms do not cause ravenous hunger. So my parents were wrong about the reason for my appetite as a child. Yes, they were. I definitely think they were wrong. Some rodent biologists, that is, human biologists studying rodents, got to wondering what parasites did do to the appetites of their favorite mammals. Researchers treated African ground squirrels with ivermectin and fipronil. What is
Fipronil?
Fipronil. Treated squirrels stopped itching and spent more time eating. Surprisingly, they did not gain weight. Parasites had depressed the squirrel's resting metabolic rate. Parasite-free squirrels had to eat more to maintain normal weight. Despite not gaining weight, treated squirrels did have a lot more babies, so they were healthier. Now, when Merck is going to donate 1 billion doses of ivermectin... No, wait a minute. Now, when is Merck going to donate 1 billion doses of ivermectin to the squirrels of the world? And then there are several references given.
Fipronil is an insecticide.
All right. I see.
So with the treatment, they didn't gain weight.
Right.
They had to eat more.
But they ate more to just stay even.
It's interesting.
It is.
Why would the parasites depress their resting metabolic rate?
No idea. I have no idea because if I were a parasite, I'd want the host to eat more so I could have more too.
So the papers are energetic costs of parasitism in the Cape ground squirrel, and parasite removal increases reproductive success.
Right.
That would make sense, right?
Right,
in a social African ground squirrel.
It would.
Well, actually, the parasite doesn't want to interrupt the reproduction of the squirrel, does it?
You're asking human-related questions to a non-human situation.
But we are humans, Dickson.
To want or not to want, I don't think those are proper phraseology.
It's not a good outcome. I know it isn't, but you know what I mean.
I think I know what you mean.
Because if you inhibit the reproduction of the host,
what would
you don't
happen
have any
if
more
the
hosts.
Parasite,
right?
but for that particular parasite, they get their shot at that host, and that's that.
The next email is from Jonathan, who writes, Dear Doctors Racaniello and Despommier, thanks for the wonderful job you do with TWiP and TWiV. I appreciate all the time and effort you put into the podcast and look forward to each new episode. I was wondering why you haven't yet given much, if any, attention to bedbugs on TWiP. They are arguably the most important human parasite in the U.S. and growing at an alarming rate. 20 years ago, bedbugs were extremely rare in the U.S., but now cost the economy an estimated $3 billion annually. The emergency department where I work is forced to close and decontaminate a main treatment area room approximately every 3 days for an average of 17 hours at a time due to a bed bug being found on a patient. While bed bugs are not vectors of human disease, they can cause itchy rashes and less commonly asthma and angioedema. Bed bugs are also associated with depression, social isolation, suicidal ideation, anxiety, nightmares, and other mental health concerns. Of all the parasites you reasonably could acquire in the U.S., bedbugs would have to be the least desired given their itchy bite, social stigma, difficulty with diagnosis and treatment, and the time and costs associated with eradicating them. The CDC neglected to place bedbugs on their list of the top neglected parasitic infections, yet I can't think of a human parasite which more adversely affects the health and well-being of Americans than bedbugs. Any thoughts? Thanks, and keep up the wonderful podcasting.
Well, we did have Dr. Gwadz on, and he did discuss bedbugs.
He did?
He did.
A lot?
No, he didn't devote the whole issue to it, but he did discuss them. And so we could, I don't know any other bedbug experts, to be honest.
Maybe Bobby does.
Maybe.
Jonathan is an assistant professor at Case Western Reserve University.
Right.
I'll take the next two, Dickson.
Okay. Jim writes, nothing new here, but it's in your region, so just in case it's of use. And he lists a website for ticks in New York.
Ticks are abundant in New York and region despite the cold winter.
They sure are.
From Cornell University.
They're very abundant.
Despite the cold winter, ticks are abundant in New York State and surrounding areas. The persistent snow cover helped insulate overwintering ticks in the leaf litter.
Yeah.
Black-legged ticks.
Yep.
Neat. It's a nice article. So.
It is, okay. So Heather writes, Dear Doctors R and D, just a quick clarification. All invasive species are exotic species, but not all exotic species are invasive. Invasive species generally cause ecological, health, or economic problems. Dr. D's beloved trout are exotic across much, if of their current global range. In many areas where non-native trout, such as rainbows, have been introduced, they are not considered invasive, but they are exotic. Large and smallmouth bass are in a similar situation. However, brown trout in public waterways in the U.S. are considered invasive. Pike in Alaska salmon rivers are one of the most recent and serious human-caused aquatic invasions. Regards, Heather M. Giddings. Now, it's also true for Yellowstone Lake, by the way. There's an invasive there, the lake trout, which may have been introduced during the 1988 fire, when they were scooping up water from other lakes and dropping it on the forests that were on fire around Yellowstone Lake. They may have introduced lake trout this way. And they ended up eating almost all of the cutthroat trout, because that's what they eat. They eat other fish. And so they had a wonderful meal of Yellowstone strain cutthroat trout. And so now they have contests on Yellowstone Lake to try to catch as many lake trout as you can, to try to get rid of, well, they'll never get rid of all of them, but to keep their numbers down.
The next one is from Scott.
Yep.
Sarcoptic mange is having a significant impact on the current wolf population in the Yellowstone area. In addition, mange is also affecting the fox and coyote population in the Front Range of Colorado, and I suspect the problem extends even further. I think people would enjoy listening to a program on mange. Where did it originate? Is it the same scabies mite humans can get? I also have photographs of fox and coyote with mange you could post on your website.
Well, thank you.
Do you know a mange expert?
Well, once again, I don't, but Bob Gwadz would know that person, so I could get in touch with Bob.
Can you do that, Dickson?
Yeah, I can do that. Of course I can do that. I'd love to do that.
Tell him we'd like a show on mange and also a show on bed bugs.
You got it.
Do you know what mange is?
Yes. Home on the Mange.
That could be the name of that title,
That's
right?
true.
Episode.
Greg writes, Greetings, professors. I am a soon-to-be first-year veterinary student and longtime lover of parasitology. I just discovered TWiP, and I am working my way through the early episodes where you discuss, among other things, the discovery of Trichinella spiralis. You mentioned Paget and Virchow, but do not mention Joseph Leidy. No, because, okay, fine. Leidy, a noted anatomist, paleontologist, parasitologist, and overall polymath, and one of my favorite scientists, is often credited with the discovery of encysted worms in pig flesh. In 1846, several years before Virchow's work, Leidy made the connection between undercooked pork and human trichinosis, that's true, and advocated for the thorough cooking of meat before consumption. Apologize if you touch on this subject in a later episode. I'm working through them as fast as I can. Big fan of the show. Greg, I agree with you that Leidy is a shining light in the history of trichinellosis and made the connection between the disease it causes and humans eating undercooked pork. Virchow worked on the life cycle And, of course, Paget discovered the worm to begin with in 1835. So there is a lot of early work that I didn't discuss when I was discussing the life cycle in the early days of trichinosis. So there's lots of room for including these wonderful historical vignettes in future episodes. So thank you for pointing that out.
I think we should record that whole episode again, Dickson.
Sure. Why not?
All right, that'll do it for email today, Dickson.
Okie doke.
This episode of TWiP will be found on microbeworld.org/TWiP and also on iTunes. And if you are new to TWiP, go over to iTunes and leave a little comment or a star rating. That helps to keep us visible. We need more subscribers.
Do we?
Yeah.
Oh, gee.
Many more.
Wow. In the billions and billions?
I don't need billions. We could use tens of thousands more.
Oh, okay.
I mean, we have lots of great listeners, but we
do.
want more because we
always want more.
Need to educate the world about parasites. They're pretty cool, don't you think?
I love parasites. I just love them.
And send your questions and comments to TWiP, TWiP at twiv.tv. Dickson Despommier can be found at verticalfarm.com. Thank you, Dickson.
You're welcome.
I appreciate your participation and erudite contributions.
It's my extreme pleasure to do so.
I'm Vincent Racaniello, and you can find me at virology.ws. The music you hear at the beginning and end of TWiP is written and performed by Ronald Jenkins. You can find his work at ronaldjenkins.com. You've been listening to This Week in Parasitism. Thanks for joining us. We'll be back soon. Another TWiP is parasitic.